I'm a clinician, so I'm not really going to get into the basic science, biology, and some of the nitty-gritty details. So I'll stay pretty broad. But if there's any issues that need clarifying, please just jump in, um, and we'll have a discussion. All right. So up front, what I'm going to talk about is a clinical trial that was funded by the California Institute of Regenerative Medicine, CIRM, which is a major player in the gene and cell therapy arena. And I note that Jeff Sheehy is actually in the room, and Jeff was a major player within CIRM on their advisory board, and I do believe had a role in getting this funded behind the scenes. So thank you, Jeff. Anyway, so I am going to present on behalf of my colleagues, including Radha Betty, who's a clinical trialist at UC Davis. This clinical trial was actually not done at UCSF, it was done at UC Davis. I'll tell you why. The technology was largely developed by Kim Anthony Ganda in collaboration with Boral Drupalich, both at Karen Cross at the time, and some of the cell processing, Jane Weiss. So it was a team effort, and I'll come back to that at the end. All right. Um, so most of us know that one of the major initiatives within the HIV cure space is to use immunotherapy to enhance the capacity of the immune system to control the virus. This is not really a true cure, right? This is generally referred to as a remission. But if we can achieve it, I think it would fit a major need within the treatment paradigm. And we're optimistic this can happen in large part because the immune system can do this. We know because based on the studies of elite controllers and now post-treatment controllers and now even post-interventional controllers, all these different people who are out there controlling virus. At the end of the day, when you look deep as to what the mechanism is, it's probably multifactorial, but central is the capacity of the CD8 T cell population, the killer T cells, to seek out, identify vulnerable parts of the virus, and then kill the cell that actually has that virus. Okay. And so the question is, how best can we go about in inducing this in people who don't actually have these naturally? And I would say 50% of our effort in the cure space is focused on developing these types of therapies. Now, complicated slide. But let me just sort of give a background on where we are. We have been trying to generate an immunotherapy for an HIV remission since the mid-1990s. The first clinical trial I was involved with was a therapeutic study of a vaccine, VaxGen, back in the early 90s. And we've done 50, maybe 100. Someone has to do a count. I'm sure Richard Jeffries would know the answer to that, how many of these studies have actually been done, but I think it's been dozens and dozens of clinical trials have been conducted over the past 3 decades to try to achieve sustained control of the virus. They've all failed. Up until now, we have 4 or 5 small pilot studies, all of them flawed, but all of them saying the same story, that if you give broadly neutralizing antibodies during either when the virus is around, you somehow induce the immune system to control the virus. So that in the study that we did of 10 people, 7 kept their virus around 1,000. 10 to the third is 1,000. In the Rio study, same thing. About a half of people who got these antibodies kept their virus around 1,000. Same thing even in people with chronic infection in a recent study done by Marina Kesk. So there's several of these studies now, very consistent. Broadly neutralizing antibodies induce partial control. It's not a cure. It's not quite where we want to be, but 1,000 is pretty good. Mechanistically, we're not entirely sure how this happens, but it does appear that there's some magic that occurs when these CD8 T cells bind to the virus and these complexes induce a CD8 T cell response. Okay, so further confirmation that at the end of the day, it's the CD8 T cells that we're interested in. Now, there have been a lot of studies that have done deep dives into the mechanism of this post-BNAb effect, trying to figure out how antibodies are inducing control. And it seems that the key thing, as I just said, is that these antibodies are inducing indirectly the generation of CD8 T cells that attack HIV, but not just HIV in general, can actually attack the vulnerable parts, can kill but very importantly, and this is kind of new, have the capacity to proliferate, have the capacity to expand, have like stem cell-like activity so that when the virus is seen by the cell, which is like kind of at rest, pounces like a cat and can basically immediately kill the virus before it spreads. So the question is, if we can't generate these cells with a therapeutic vaccine, and if we can't really get as much out of a BNAb as we want, what next? And I think what next is actually using gene therapy and asking the question whether we can just build these CD T cells directly, okay, like they're doing in the cancer setting. Now, that gets me to this focus here, CAR T cells. What are CAR T cells? So in generally, most of the CAR T cell work is in cancer, all right? Um, and the target cell here is the cancer cell. The CAR T cell is basically your CD T cell, your CD4 T cell that's been gene modified. The cells have been taken out of the body, sent to a lab, genes been inserted into the chromosomes of those cells. Now those cells express the CAR. And what is the CAR? The CAR is on the cell surface, it's like an antibody that binds to the virus. And the antibody on the cell surface is linked into the cell through a gene that stimulates the cell so it proliferates, and CD3, which actually helps the T cells do what it needs to do. So essentially, you're gene modifying these cells so that they actually can target cell surface proteins, either cancer proteins or in the case of HIV expressed on the cell surface. All right. Now, CAR T cells, as I'll mention, were first developed for HIV, then didn't work in the 90s, then exploded in cancer and is working now in lymphomas, leukemia, multiple myeloma, and even some solid cancers. But it's a massive amount of work going on using these CAR T cells in all sorts of diseases, with a massive explosion of interest in autoimmunity, diseases like lupus, and a variety of other autoimmune diseases. Why is that important? Because now the CAR T cells have left the cancer field, where the risk benefit of CAR T cells is quite different than in HIV, into autoimmunity, where people generally don't have fatal illnesses, like with HIV, but yet can benefit from these cells. And that's really important because now we're having proof of concept that you can do this outside of cancer. And there's a huge now effort trying to come up with ways to do this safer because the population is expanding. So there's a huge revolution going on with CAR T cells across medicine. But let me go back and tell the history because I think it's really important that everyone is aware of this in general. As it happens, the first CAR was actually discovered by mistake here at UCSF in the lab. Someone trying to figure out how T cells work. And then it quickly became clear that, well, these things, if we can make these T cells express CARs with a gene therapy type approach, well, we can do interesting things. Most of the money that went into the development of this technology was HIV money. Tony Fauci and the NIH invested heavily in this late 80s, early 90s. And so when you hear about CAR T cells, you should remind people that they need to thank the HIV investments, just like they should thank us for the COVID stuff and everything else. An important part of the story. Anyway, there were a series of studies done in the 1990s, and we did the first randomized study of a CAR T cell back then. It didn't really work. And then treatment came along, and everyone left HIV and went into cancer. Now they're all coming back. But again, this concept about how HIV is a major driver of innovation outside of HIV is a really important story. And the CAR T cell story is just one story. Anyway, let's get to the product that we're talking about today. This is called the LVGP120 dual. I'll just refer to them as these dual CARs. Dual means there's two separate CARs, two parts of the envelope. What are these? What are the cells that are made? Well, these cells actually express on the cell surface two receptors, one that binds to the CD4 binding site of HIV and one that binds to the CCR5 binding site of HIV. Okay. Basically, when the HIV particle in an infected cell gets expressed on the surface of that cell, the CAR T cell will bind to both parts. It's like combination therapy and kill the cell.
Eve, those two binding sites, those are basically the doorknobs that HIV uses to get into the cell?
No, it's like the door. It's not the doorknobs to get in. So it's the HIV coming out. So the cell
Okay.
has got HIV. It's infected and starts making particles. And those particles, as they come out, the first thing that comes out is the envelope proteins on the surface of the
Got it.
cell.
Got it. Got it.
And that's when the CAR T cells pounce and kill.
Got it.
Which treatment doesn't do that, by the way. Treatment does not kill, right? Treatment allows the
Right.
virus to come out. It just prevents it from going to the next cell.
Got it.
All right. And in mouse models and everything else, it's actually appears that these cells have these properties that we're talking about, the ability to target the right part of the envelope and the ability to proliferate stem cell stuff and kill. So we think they got the right characteristics.
Mm.
All right, here's a study. This was done again. It was supposed to be done at UC San Francisco, but this was happened during COVID, and UCSF at the time said they didn't have the capacity to support the study. And so we ended up doing the entire clinical trial under Merdad's leadership at UC Davis. So the study was designed to be a safety study. Okay, this is the first time that there's been several CAR T cell studies in HIV, and there's several going on now, but this was the first time that this product had been studied. So we did a traditional phase 1 dose escalation safety study. In the first cohort, which we didn't really expect the cells to do much, there was 3 people. They did not. Okay, let me back up. Generally, in the CAR T cell business, it's generally accepted that in order for these T cells to engraft and persist and work, you have to quote unquote make space in the bone marrow. And you do that by giving people low doses, either very high doses in cancer or in our setting, very low doses of drugs that can actually kill temporarily the lymphocytes in particular, lymphodepletion. And we use low doses of cyclophosphamide because this has generally been, at these doses, been used in a variety of diseases for many years, including a lot of the autoimmune diseases. Generally thought to be, not benign, but not as safe as not as unsafe as other things. I'll come back to that because this is where we got into trouble with one person. So anyway, we gave this low dose cyclophosphamide, which we've done our group in about 100 people in the past. We've never really seen much except a drop in lymphocyte counts that are transient. So anyway, the study is the first cohort got none of that and got a very low dose of cells and stopped therapy. Second cohort got the low dose conditioning and a low dose of cells, stopped therapy. Third cohort got low dose conditioning, high doses, standard dose of cells, and stopped. Okay. Now, the study design is a little complicated here in terms of what happened, but we enrolled 9 participants, but we allowed people in this first cohort to re-enroll in the second and third cohort. So we ended up having 11 infusions. 2 didn't stop therapy, only 9 stopped therapy. It's a little bit of, at the end of the day, 9 people got their infusions and stopped therapy. All right. So, and the idea here is that, if someone comes in, they're on treatment, viral load's undetectable. This is what typically happens when you stop therapy. The virus, after several weeks, rebounds. Okay. We know that. We've seen it over and over again. The idea here is that you give the CAR T cells at the time of the interruption and that they take a few days to expand and to become functional. And at around day 5 or so, this axis is off, usually around day 5 to 7 post-ART, you expect these cells to now become very functional. They will block this rebound. And if you can block the explosion of the HIV, the cells can, in theory, get ahead of the virus and keep it under control. That's the theory. All right. Now, we have no control arm. And I don't think for these types of studies, a control arm is necessary. This is controversial in the field. And I don't think a control arm is necessary because we really have such pristine data that tells us what happens when people stop therapy. And what happens is that typically at around, 2 to 3 weeks, the virus rebounds robustly. But every now and then, someone rebounds and their virus goes down below 1,000 safe. But usually 90% plus or minus of people, when they stop therapy, the virus explodes. They have to go back on therapy. But every now and then, you get people control at a low level. Sometimes you get people control undetectable, but that's very rare. But anyway, so this is the background. So what happened in the study? By the way, so the eligibility criteria, generally healthy people, no comorbidities, on treatment, virus under control, higher CD4 counts, and so forth. Pretty typical stuff. We did enroll people who started therapy early and late, and that's going to be important in how we discuss the data. This is San Francisco, so this is not too much of a surprise to everyone, but this is an unfortunate truth that we have to deal with in San Francisco when we do these studies. We just have the hardest time enrolling women, and so we ended up enrolling, even at UC Davis, we ended up enrolling 11 men, all cisgender, but we did have a diversity in terms of some of the ethnicity background. But this is all older men, typical for the types of people we typically enroll in our settings. People had been in long-term treatment for over a decade for the most part, and their CD4 counts were generally above 500, and they had high CD4 nators. So generally a healthy group. Okay. First cohort. Again, we didn't expect anything to happen, and nothing really happened. Okay, so 3 people with chronic infection came in, got in this group, they had no conditioning. They got the low dose cells. They stopped therapy. This person rebounded around the 3rd week. This rebounded around the 3rd week, and then around the 2nd week. The virus rebounded, went up to high levels. People went back on treatment. We then asked the question, well, why did nothing happen? And it turns out that when we look at the frequency of cells that had expressed the CAR, that actually had the CAR and had been successfully gene modified, the number of copies of the CAR per million cells in the blood, essentially 3 people, we couldn't detect anything. And 1 person, it was low levels and bounced around. So as expected, it was low dose of cells, no conditioning. We didn't see much CARs that persisted, and we didn't see much of an effect. Cohort 2, again, got low cells, but this time the conditioning. First person, again, chronically infected when he started therapy. Virus sort of rebounded as typical. The next person, though, is different. So this person started therapy during early infection, febic 4. So several weeks, not super early, but not late. He's HLA-B5701 negative, so not destined to control based on their background genetics. Indeed, stopped therapy. We documented that by looking at drug levels, and has controlled for about 2 years now, with occasional blips. But the virus has largely been below the level of quantification, but detected, but basically undetectable for 2 years. Very unusual, but it does happen. So we can't say that this is the CAR-T cells. But then it happened again, and that's the coincidence. Is the math of this happening twice is it's not impossible, but it's very unlikely that this is simply just luck. Second person, same story basically, and he's now about a year out, undetectable. And these guys, when we looked at the number of CARs in their blood, because of the conditioning, as expected, they had more CAR-T cells, low levels, but more. But they didn't persist. They actually generally went down to low levels. I'll come back to that. Third cohort got the conditioning and now the standard dose of cells. Again, 1 person chronically infected, stopped therapy, typical rebound. Then one person who started therapy again, phoebic 4, early, didn't keep the virus undetectable this time, but rebounded and kept it around 1,000, which is kind of like what we see with the broadly neutralizing antibody story. So appears to have had some partial effect, but was not sustained for more than a few months. But this looks again, this can happen in the absence of anything else. But again, this is the third. 3 out of 3 people who started therapy early had some control. And then lastly, we had a person with chronic infection, whose CD4 nadir was lower than the rest of the crowd and who was older. And he actually maintained a virus that was quite low for about almost 3 months before he went back on therapy. And he went back on therapy in part because of other issues, not because of the virus that I'm going to come back to. But his virus actually never rebounded above 1,000 below before he went back on meds. So 4 of 9 people have evidence of control. 3 of 3 who started early had evidence of control. And in 2 of them, it was really tight control for a year to 2, which is, again, unusual. We asked the question, if you compare time to rebound, if you look at all 9 people and look at the time to rebound, which is an early marker of how these cells may or may not be working, just preventing the virus from rebounding, it was basically substantially longer, suggesting that across the entire cohort, we're seeing some biologic activity. Another confirmation that there's something going on here. Okay, now the big issue is safety, right? This is a study focused on safety. Now, when this study was put together, 99% of our focus was on the toxicities that you see with CAR T cells in cancer. There are 2 big ones. Okay, there's neurotoxicity. This is where a couple weeks after the CAR T cells are administered, people get neurologic changes, mental, their alertness goes down. They can often be admitted to the hospital. It's reversible, but it's not something you'd ever want to see. So that's why we went very careful in the study, and we monitored people daily, more or less, for a month. And we didn't see it. And we also didn't see cytokine release storm. This whole concept that when you give the CAR T cells, and you cause all these cells to die, you get an inflammatory response that can make people sick. We didn't see that either. Now, several years later, we now have learned, mainly from what's happening in the outside of HIV in the other fields of medicine, where CAR T cells are now being used outside of cancer, that these toxicities are related to the overall burden of the disease that we're going after. And the way it was explained to me is like with a cancer, it's like a basketball in terms of how much tissue is there. With autoimmunity, with lupus, it's like a tennis ball. With HIV, it's like less than a BB. How much of the cells are there that have HIV in it? And so we're not really, the more that what we've learned is that it's how much cells are being targeted and killed leads to neurotoxicity and cytokine. So we didn't see it, and now most people don't expect to see it, which is really important because this will allow us to, I think, move forward. So we didn't see any real toxicity related to the product, but we did, and people have heard this, had a unexpected, that last person that I mentioned, the older guy, his CD4, his lymphocyte count, neutrophil counts dropped dramatically. And we're still trying to figure out why. We think he had some pre-existing bone marrow reserve issues. The neutrophil count came right back up, and the lymphocyte count is actually coming up, but very slowly. And so this can happen. We've learned that this does occasionally happen outside of HIV, more often in older people. But again, in the cancer setting, this is the cancer CAR T cell people that we're working with are very nonchalant, saying, yeah, well, this just happens and this is part of the will actually make the CAR T cells work better. But of course, in the HIV setting where people are generally healthy, was a significant adverse event. But the participant's doing well. He feels fine. And his numbers are coming back slowly.
Steve, excuse me, do you think that the general health, better health of people without cancer is contributing? I mean, the fact that this person was older might tell us, what we might need to use for an inclusion criteria.
Yeah, for this to go forward, if there are two options. We got to come up with ways not to use conditioning, or we have to come up with ways to screen people better. And I prefer the former, and I'll discuss where that's going. Um, but the issue here is age,
Yeah.
right? This is the big factor. He was 60, he's 68. Um, and I think that is the so we may in the future need to enroll a younger population until we figure this out. Um, yeah.
It's important to know that because we've been on part of our journey has been in increasing that age limit. So, if this all of a sudden is a lower age limit, that's great. We want safety. It's good for us to know so we can let people know that's the case, why this is happening. So,
Absolutely. No, I'm a big proponent that we should be doing most of these studies in older people. But this is not, I mean, we got to think about this because
yeah.
this in hindsight is something we probably could have predicted. It's unlikely that the CAR T cell product, which was our focus, would have more toxicity in older versus younger, but the conditioning, which again, we weren't too worried about because we've done this before, in hindsight, we could have predicted this. Um, the other big thing, of course, is the treatment interruption, but we know how to manage that and generally didn't have any issues, and the participants were all highly engaged, and it's really a very sophisticated group, and most of them understood issues of treatment interruption and transmission and so forth, and so generally that wasn't an issue. Um, I can end there. Let me just talk a little bit about the mechanistic stuff here, because the riddle here is why would this work if the cells are at low levels, which they were, and disappear after a few months, as they did. Okay. One, this could have nothing to do with the cells. These people were destined to be post-treatment controllers. Unlikely, but possible. Two, the cells are not in the blood because that's not where the virus is. They're in the tissue fighting the battle, so we don't see them. Three, the amount of virus in the body is quite low. So therefore, the number of cells that you need to control that virus is quite low. And this is something you see in cancer. If you have a lot of cancer, you end up with massive number of cells expanded, and it's usually proportional. Four, we may end up not needing, if you have T cells that work for the first few weeks of rebound, and you slow down that rebound, and you prevent the virus from exploding and causing all sorts of mayhem, then the immune system actually can figure out, well, how to respond to the virus effectively. We personally think that's what's happening with the antibody studies. This is a big focus of DARE. Targeting that early rebound, preventing the explosion systematically, letting the immune system figure it out. And then finally, they see this in cancer too. And there's that the anti-cancer effect of T cells persists for years, long after T cells are sort of gone. And they think that CAR T cells are educating through various mechanisms. So we're trying to figure this out. So Linda, you asked up front, we need to figure out why it's working in some and why it's not working in others. And so this is sort of where using the biologic specimens from this study and other studies, we're going to try to figure that out.
Yay.
All right. Now something I'm passionate about. All right. Why are we doing this? And will this ever be useful? All right. In my opinion, not shared by everyone, and people do get annoyed when I say this, the reason we're in this business is not because treatment is fundamentally flawed. Treatment's amazing. It works great in most people, almost everyone these days. It's not the drugs. It's the fact that people have to engage with the healthcare system forever. Okay, that's the problem, I think. And there are a lot of issues that get in the way. Individual levels, public health issues, global funding, whatever. There are 1,000 different reasons why someone cannot, from the time they have HIV to the time they die, stay involved in a healthcare system. So if that's what we're trying to fix, which I think it is, then the cure is going to have to be something that works in a population that's not engaged indefinitely. Okay. It's going to have to be scalable. It's going to be safe. It's going to have to be short term. It's going to be affordable. Right. And these things are not affordable. We can cure sickle cell. There's a lot of synergy between HIV and sickle cell. They're both very common in sub-Saharan Africa, where the focus is these days of both of the research agendas. They're both chronic diseases with significant morbidity, significant stigma, early mortality, at least particularly with sickle cell. And they're both amenable to gene therapy cures. And we can actually now cure sickle cell with gene therapy, very similar to what we're doing in HIV. You take the cells out, send it to a lab, you gene modify it, you put it back in. Problem is, these cures cost millions of dollars, which is nuts. But that's why these cures are not having a big impact. There've been, I don't know, a few hundred people I think cured because of cost. So, and CAR-T cells cost about $500,000 just for the product. So this is never going to fit the unmet need, and it's never going to have much of an impact if we don't fix this, right? What we desperately need are one-shot cures. Now, I just say that is because the field is really shifting, the CAR-T cell field, from this concept that you have to take cells out of the body, send them to a lab, bring the cells back, give people chemotherapy, infuse the cells, to in vivo, where you basically, just like we had with the COVID vaccines, you do an injection in the arm and in the blood using the same technology of the COVID vaccines, these LNPs that deliver a gene to T cells. And you can do this without conditioning, without chemotherapy, without cell processing, cost of goods $1,000, $2,000. And so the reason why I think that the CAR-T cell, now that's a decade away. And we have to figure out whether or not it makes sense to pursue this. And I think that these pilot studies that we've done, that are being done in China, that are being done at UC San Diego, being done at Penn, they're going to be helpful to inform to what the only reason it makes sense in my mind for this to be happening in the HIV space is that there is a viable way to do this in a scalable manner. In conclusion, we found that the product itself was safe, which is a big deal. But the conditioning regimen certainly poses issues. We did see that the conditioning helped with the expression of the CAR. We saw post-treatment control in more people than we expected, and very tight control for a year to two, unusual. These data provide, I think, proof of concept that there are now viable pathways to one-shot cures, but of course, it's going to have to be optimized. There's a large team. Some of the key people here I mentioned early on, and I just want to clarify that a lot of this stuff is being done in the context of a huge global effort to get gene and cell-based therapy technologies to sub-Saharan Africa, and we're involved in that and through a variety of different forums.
Can I say something really quick about the cost, Steve?
Oh, sure, Jeff. Hey.
just about carrying cross and its unique model. I think Linda's familiar with this.
Yeah.
Through technology transfer and through process optimization, there and partnerships with other governments, they are delivering CAR T cells through the public health system in Brazil at a tenth of the cost. So it is a business model that is not Gilead. It's a business model that's been is one of the most powerful things in their application for me at CERM was that they went in with the dedication to make this available, to not hold on to technology, to look at scalable processes that can be done, but looking at mobile gene modification, all sorts of things to try to share the wealth instead of hoard it and pay shareholders. And they're a public benefit corporation as opposed to a shareholder driven one. That one little element I think is important because it's obviously is complex and difficult technology, but it is also, there's a commitment to making it available to people all around the world.
Jeff, I just wrote down price, as Steve was finishing up. And the problem is that we're not Brazil, and I think that we have to realize that the government is out to get HIV at this point. So I don't know what they're going to fund. We don't know, what's going to happen. We still don't know what's what's happening for this year. It's crazy. But the other thing is, I mean, I just don't see this government using that sort of model. They'll have these companies they're getting kickbacks from making all this money like they do in with other diseases, and then no, and five people get it. So it's really frightening to me that we're at this stupid juncture where we have to worry about that kind of stuff. So I don't know, and I think Boro's he's really his model is instead of this do everything at a shop where the people live instead of having to take it here, freeze it there, fly it here. And I mean that he's devoted, dedicated to making that work. And maybe it's a good idea that it's happening in Brazil first. I mean, it would be great if we could show that to the powers that be. It depends on who wins this election and that election, but it makes me concerned that all of this stuff is so expensive at this point in time. It's kind of scary to think about how much of it we can get done. But that's why we're here, right? So help with some of that. But Michael, you usually ask the questions to people. I see all the questions are from you. So why don't
Yeah,
you go
so
into the questions?
well, because I know the person, so I was like thinking about this ahead of time. So that's why I had a lot of
Uh-huh.
prepared questions already
Well,
written
that's
out, so I could
good.
just cut and paste,
Go
because I
for it.
stuff that I would want to talk about anyway. But and so if you also have questions, people, I just want to make sure everyone realizes putting them in the chat or raise your hand, we'll be able to answer them that way. But trying to go back to the beginning of my questions here, Steve. One of those things that you said, I've thought was interesting. You said like you know, you brought it up about getting into the tissues, and I was like, do you have any evidence that the duoCAR-T cells are reaching the lymph nodes or the gut or the brain? Like, are you guys looking at that in this study? Is there ways to look at that, see where it's going, or is it just circulating in the blood only, and that's all you've been looking at?
Unfortunately, because the study was done at UC Davis, we were not able to leverage we have
Right,
the scope machine. We've
right.
done thousands of gut biopsies. So we were only able to get gut biopsies and I think a lymph node biopsy in two people. But the,
Uh,
the, the people who matter, the controllers. So we're looking at that. But we
yeah,
don't have any data.
cool. And so the other thing I wanted to know is like, how are participants being monitored for any sort of delayed harms that might happen? Like I know you had a
Yeah,
lot of safety in there, but what about afterwards? Like, abnormal immune reactions that might happen or damage to their cells, insertion related cancers, all those
sure.
lovely gene
Yeah.
therapy things.
Off target
I,
issue.
I should have talked about that. So because you are inserting a gene in the DNA, there is a chance of causing the cell to become transformed to become a cancer cell. And in fact, this has been seen in certain types of gene therapy cells, mainly when you're looking at stem cells, hematopoietic stem cells, and using strong promoters. Here we're looking at T cells. which for some reason are resistant to becoming transformed. We know that because, HIV does the same thing in T cells, and you never get lymphomas as a consequence of the HIV integration. But it's an issue, it's a concern. It's because we're not using stem cells and we're not using strong promoters, it's a lower concern. But we follow people for 15 years, and there is a registry. And if we see a lymphoma, you do studies to see if the CAR is inserted in the right place in that lymphoma. And there have been a couple cases. I mean, there's been 10, 20,000 CAR T cells administrations. There are a couple of potential cancers, but they're controversial as to whether it's real or not.
Mm, mm.
Real
So-
and that not caused by the actual procedure, right?
Not caused. There are definitely, again, with certain types of stem cell therapies, clearly gene insertions are dangerous. But in your CAR T cells, where you're looking at inserting a CAR in T cells, not stem cells, there's a couple of potential cases, but they're being litigated as to whether it's real out of 10, 20,000 times. So the
And
risk
so-
is low, but not zero, and so we're monitoring people.
So I wanted to ask about the first 3 participants that had no detectable CAR T cells after the infusion. What have you
Right.
learned about why the cells failed to persist at that moment, and how much of the later persistence was caused by the I can never say the cyclophoma, the little conditioning, rather
Cyclophosphamide, yeah.
than the CAR T cell product? I'm sorry, I can't say that word. I should
Um,
practice.
Cyclophosphamide. But anyway,
Cyclophosphamide. Okay, thank you.
believe it or not, the thing I find interesting about HIV research that's different is that in the HIV world, whenever something happens, we always ask the question why. That's just the way with this. We're all hardwired, and it's always about biology and mechanism. That's not the way it exists everywhere else. They have been doing conditioning in cancer studies forever
Mhm.
because it works. But no one's ever actually explained why it works. And I ask people, why do you do this? What does it actually do? And they go, oh, it makes space. Well, that doesn't make any sense.
Right.
And some people say, well, it prevents it suppresses the immune system from killing the cells. We don't know why the conditioning is needed, but it clearly is if you want to get better persistence.
Mm.
It's a difference between the kinds of doctors.
I don't know why it is. I think we're in the 90s, we all got used to asking why and led to good things. So we keep doing it.
That's because that's when we came in.
So I also had a question about like how you could actually determine whether the duoCAR-T cells were the ones causing the control rather than early treatment or the natural post-treatment control that some people have. Is there a way to suss that out from what you've done in the study?
Nope. The only way to suss that out is a randomized control trial.
Yes.
Okay, that's what I was.
Well, we'll have that too, right? Hopefully.
Eventually, but this has to be big and they're costly, and I'm not sure when that's going to happen.
I also had a question about like, do we know if HIV can mutate so that the duoCAR-T cells would no longer recognize it?
Sure. HIV is pretty clever in this regard. And the envelope in particular, the dual CARs are attacking the envelope protein, which are very variable. So this is definitely an issue. It definitely can happen. We haven't done those studies.
Okay. I know the Korean had a question. I wanted to read it off, and I'm looking to see where it is. Oh, do you know how the duoCAR participants feel about the product, the conditioning, the safety profile, etc.? She wanted to say that she thinks it would be interesting, of course, being a social scientist, that we try to get some retrospective data on that.
Yeah, as I recall, Corinne, we were going to do that when the study was at UC San Francisco, and then I think we had trouble getting it implemented at UC Davis, which is unfortunate because the experience of any gene therapy study is quite different than anything else because there's a whole slew of issues, right? Mainly regarding safety, short term and long term. We weren't able to sort of quantify that in this pilot study.
Mm. Mm. I also had a question. You kind of brought it up when you were talking about the types of people that you got to enroll in the study and how that's a problem in San Francisco and maybe in UC Davis. But how representative do you think these participants were in terms of all the people who get HIV around the world, how representative do you think they are of this, or do you think that that's a problem?
I think they're a very important population, but older men with generally HIV. But they're only representative of older men in urban settings with HIV. Yes.
Right,
So this is not generalizable to what the epidemic looks like in the South
Anywhere.
or but it's an important group of individuals that doesn't get the attention they deserve, I don't think. But anyway, it's a separate discussion.
right. Well, I also am curious about, specifically women, because all this evidence of that there are sex-based differences in what happens in terms of HIV, and so just
Sure.
not having any women in that's what I wonder if there's anything that we should be looking at there, and like how would we maybe even enroll women in these difficult studies that are even difficult just for other people.
I think that's actually where I would if Korean has the capacity and the bandwidth, I would start there.
I agree.
Not asking people how they felt during the clinical trial,
Mm,
but why certain populations are very hesitant to go into these studies.
hmm
And you talk, we try to recruit women, and the younger they have HIV, it's a different population. Much more of them are black and have a lot of socioeconomic baggage as a consequence, lots of poverty, domestic issues, children. I mean, a lot of logistical feasible issues, but there's also a distrust, particularly around gene therapy, that exists in a lot of populations that I think we're going to have to figure out.
Kareen's writing into the chat that we were able to get 2 women in the CMV CAR T cell trial in San Diego.
Wow, that's wonderful.
Yeah. And then Jeff, you wrote Henrietta Lacks. Care to expand upon that?
Well, just that's a classic example in the African American community of why people
Don't trust.
don't trust the system,
Yes. Yes.
not
Uh,
just Tuskegee.
But too, you also have to look at, and I could scream about this. I've been screaming at every antiviral approval hearing about enrolling women and people of color. You have to see why they're not doing it. Do they need childcare or do they need support? Do they need to know more than they know now? I mean, and as far back as the Merck real Merck study and the one that Tech did, for women, the Grace study. And they said that it's not only that you have to get people in, but you have to do things to make them stay in because they end up leaving because they have all these other life problems to deal with. So I mean, it's a multi-factor, just like everything else. But I mean, I think that what we've always tried to do was get the drug companies to put up some money for it so
mm,
that we could help people, especially women who take care of everybody but themselves for the most part, be able fit, just reasonably and physically to get in there when they have all these other things going on.
hmm Kareen, I saw you had your hand up and then I saw you in the chat, but I want you to come off a mute
Yeah.
and
So I think
share.
I agree with Steve. It's important to also know what the barriers are, but we've done a lot of work on this, trying to find out what are some of the barriers and facilitators. We also need to be able to implement those recommendations. So I do think there's still a lot of implementation and implementation science that needs to happen alongside these clinical trials, particularly if participants are going to need 15 years of follow-up. Jeff, Michael, myself, and others are going to be starting a US-based survey soon. So hopefully we'll also find out about like the mistrust and distrust around cell and gene therapy and how we can counteract some of that. So I think both the community-based work and work embedded as part of trials is necessary. Thank you.
And Siggy made a comment in the chat that in Germany there's quite a reluctance from the authorities to approve first-in-human studies for women. And then Jeff responded, thalidomide.
But I think it's about having babies and all that sort of stuff. What do you think is driving that, Siggy?
Actually, I have no idea because the interesting thing is that the person that denied a clinical study, first in human study with a gene product, the person who denied it was a woman. Whereas the ethics commission, they approved. They said, "Oh, it's great that you're thinking of women from the very beginning," you know. But the authority said no.
So I just launched the poll just so people can let us know what they think of what we've learned so far today.
But while I'm on the mic, I have one question that bothers me. They always say there is a 15 years period where people are followed. But what happens when the company who does the trial, or the organization for that matter who does the trial, doesn't exist in 15 years' time? Who is going to do the follow-up for these patients?
Yeah, these are unfunded mandates. And the company is not, this is the academic institutions that are basically forced to do this. And there's no such thing as 15-year funding for this thing. CERM isn't funding this, and the NIH isn't typically funding this, so the institution has to do it.
Should
We
we
have
be requesting
healthcare.
that the FDA create a registry or something like that everybody can participate in instead of leaving it up to the institutions? Some will
Only
do it
four,
better than others, if at all, right? So
only 4 people left at the FDA, you know?
that'll
The
change.
academic institutions are generally got the resources to do this. But this is often done in private clinics now, and yeah, so I think it's an issue.
Yeah.
And there's no, and if people don't do it, there's really no one's monitoring it.
And
If
what
we
if the
had a
patient
healthcare
moves?
system, it would be a different story. And we could follow people better, but we just don't.
Diggy, you said something while Linda was talking. I didn't hear what you said.
Yeah, I said, what if the patient moves maybe to a different country or just a different city? I
Oh
think
yeah.
it's a really different question how to really implement this follow-up.
It's not done as rigorously as it should, because it's unfunded and there are a lot of issues.
Did anyone else have any other questions? Trying to look through here.
Andy Cates has one.
Andy, go ahead.
Yeah, hi. Thanks for the amazing talk. Not like you don't do everything like that, but anywho, always blessed to have a talk from you, Steve. Um, so with the ACTG, they've got A5385. And that is a study that just follows long-term people who are controlling in cure studies. I'm wondering, even given the silos that exist in the CAR-T world as far as funding, you know, something that happens in California is not going to be funded by, NIH, for example. I'm a rep on the Penn study, and that's just going to be a single site study. But if there could be, an overarching protocol that follows all of the participants from CAR T cell trials, in the vein of 5385. I think that would be something that would be great to be able to have. I just don't know how you end up with the funding for it. I don't know if you do it as maybe a I don't know, just spitballing here, but like a how sales tax works. If you're doing a CAR T cell study, there has to be X number of whatever funds that go into this organization. Any thoughts on that?
Well, I know about that parent protocol in the ECTG, and we have referred controllers to that study. We don't work in the ACTG, but there's an ACTG center in San Francisco. But these two controllers are Davis, so that's a problem. And but it's the other people, the other 7 people who are no longer who are back on treatment. Yeah, so I think that's a great idea, but and I'm wondering if the cancer networks have such parent protocols that just exist for to basically refer people. It would make great sense, but I'm not aware of one. But we're not tied that well to the cancer setting, and we probably should be.
Be great if we had a Max and Y sort of cohort group that we could, that could do this, it could be funded to do this.
The HIV cure, only a handful of centers are doing cutting edge cure research and have controllers, and non-controllers, I guess. So, it wouldn't be hard to imagine funding, there's Rockefeller and there's Penn and, there's a handful of, and there's a couple in Europe and now a couple in Africa, but there's only, this is maybe 10 centers do this work. So we should be able to collaborate better.
Yeah. But that would be a good idea to do, have somebody fund it, I mean, the government needs to fund it is what, but I don't know, what the hell will happen there.
Yeah, but
But
then the onus would be taken off of, any single academic institution. It could be a group effort and
yeah.
more sustainable and affordable for all.
Mm-hmm.
I had
That's what we
another.
did at the beginning of HIV. And, Jeff Taylor's right about the registry. It's just like a pipe dream at this juncture, though.
I had another question for Steve, and it was just because he made a comment at the very beginning when you were talking about BNABs. You said all of these studies were flawed. And what did you mean by that? How were they flawed?
Well, I mean, they're all small.
Okay.
Our study didn't have a control arm. A lot of the studies see an effect, but only if you won't go back and cut and slice and retro,
Got it.
even the Rio study, which was a randomized study with like almost 100 people, they did multiple interventions and changed the protocol halfway through. It's not that flawed, but none of
Okay.
the studies
Well, I was
are
just
It's
curious.
pretty flawed. Change it halfway through. Yeah, but we're in the beginning of this. It's all kind of
Yeah.
proof of concept stuff. So
Yes, you guys get it, but some of our critics are they want definitive studies or nothing, and that's just not the way it
Right.
works.
Yeah, no, and when I was asking that question, I was just more curious to know so I
Right.
could comment whenever I hear things like that. Like I just didn't know exactly what you found was flawed. Thank you for that.
I mean,
Does
they're all
anyone else
flawed,
have-
but if you put them together, they're all so consistent that if the data are like, if you can't see the truth in 5 or 6 studies saying the same thing, you should not be in this business. Don't
Right.
tell my friends I said that.
Luckily, they're not on this call. So did anyone else have another question? Anything you wanted to know?
Nobody else? We're going to end on our usual time instead of 2:30,
Yeah.
our old usual time. You were so clear, Steve, that we didn't need extra time. Good. And you did well, almost 70% satisfactory rate. So that's really good. Okay. Well, before we go, remember again, August the 18th, microbiome. I mean, we're very interested in the gut, where once HIV hits the gut, it goes everywhere. So we need to learn more about how that works and how to turn that off. And we'll be in touch about what comes after that. But for now, that's what's in the hopper for next month.
All
So
right.
anything
Bye, everybody.
else? All right. Thank you so much, Steve. Great to see you, Jeff, too. Jeff Sheehy. Everybody's named Jeff. Yeah.
Thank you
Thanks
all.
again, Steve. All right.
Bye.
Bye.
Bye bye.
Bye bye.